Translational Oncology Insights. I'm your host, Rittika Datta, and today we are exploring one of the most paradigm shifting frontiers in cancer biology, the reversal of cancer cells back into their normal functioning states. Now, this is not exactly science fiction, it's a cutting edge cellular reprogramming that's challenging the very fundamental understanding of malignancy as a permanent state, which it is not apparently. So, we have our guest, Dr. Sneha Pathak, a leading researcher in cellular reprogramming and cancer biology, who is a great enthusiast about this topic. So, whose work sits at the intersection of epigenetics, systems biology, therapeutic interventions. So, Dr. Pathak, the concept of cancer reversal runs counter to decades of thinking about malignancy as an irreversible transformation. Could you start by explaining the theoretical foundation that makes it a theory?
Thank you so much, Ritika, for having me. So the theoretical foundation rests on a fundamental shift in how we conceptualize cancer. For decades, we viewed malignancy through the lens of irreversible genetic damage as per the somatic mutation theory. But the emerging research evidences, this suggests that cancer is often more of an epigenetic disease, which is a disorder of cellular information processing rather than DNA sequence alterations. So the key insight is that a cellular identity is largely determined by the epigenetic states, that is patterns of DNA methylation, histone modification, and 3D chromatin architecture. Now these states are remarkably plastic. Even highly malignant cells, they retain the genetic blueprint for normal function. What's disrupted is the regulatory mechanism that interprets that blueprint. So think of a corrupted operating system running on an intact software. That can be analogy applied right now for the understanding of this situation. Now, traditional cancer therapy, it tries to destroy the hardware like chemotherapy and radiation target rapidly dividing cells indiscriminately. But if we debug and restore the operating system, we can potentially restore the normal cellular function behavior without destroying the cell itself.
That was a very vivid explanation indeed. So, I wanted to go into the epigenetic landscapes, more specifically of that of cancer cells that show profound alterations in chromatin architecture. How do techniques like Hi-C and like ATAC sequence reveal the mechanistic basis for cellular reprogramming? And what specific chromatin remodeling complexes are most amenable to therapeutic targeting for reversals exactly?
Sure. So, technology like high throughput version of chromosome confirmation capture technology, which is also called as Hi-C, and ATAC sequencing, which is actually assay for transposase accessible chromatin using the sequencing method. Now, these two techniques have revolutionized our understanding of cancer cell epigenetics by revealing how three-dimensional genome organization drives the malignant behavior. Now, the Hi-C maps, it aids us in understanding the chromosomal interactions genome-wide, at the same time showing us that cancer cells often exhibit a disruptive topology associated with domains, that is the TAD domains, which are fundamental units of chromatin organization. In normal cells, the TADs ensure that genes interact with their appropriate regulatory elements, whereas in cancer cells, we see the boundary disruptions. They create what we call it as new TADs, that is abnormal chromosomal neighborhoods, where the oncogenes they gain access to inappropriate enhancers or tumor suppressors lose their contact with the activating element. Also, the ATAC sequencing, it complements this by mapping the chromatin accessibility and essentially showing us which regulatory regions are open for business. So the cancer cells, they show dramatic shifts in accessibility landscapes with thousands of sites gaining or either losing the accessibility compared to the normal counterparts. Now, the most promising therapeutic targets are the enzymes that establish and maintain these aberrant states. So the polycomb repressive complex 2, that is also called as PRC2, is particularly a very interesting protein complex as it often hijacked in cancer to silence tumor suppressors. Similarly, EZH2, the catalytic subunit of PRC2, it kind of makes H3K27 the epigenetic marker to go under repressive mode. So H3K27 is nothing but trimethylation of histone H3 at lysine 27 position. So, consequently of this process, this particular histone, it undergoes a transcriptionally repressive epigenetic marker. So we have shown that selective EZH2 inhibitors can restore normal chromatin architecture in certain lymphomas, leading to cellular reprogramming rather than cell death. Similarly, we also have SWI-SNF chromatin remodeling complex, is a very famous, well-known chromatin remodeling complex. It is mutated in over 14% of cancers. But even when the complex itself isn't mutated, its targeting can be disrupted. So now we are developing approaches to restore SWI-SNF remodeling function to its normal genomic locations, essentially teaching cancers how to remember their original identity.
Wow, that was indeed good
Yeah.
explanation. So, transcription factor networks in cancer cells show rewired regulatory circuits. So, how do you systematically identify master regulators whose modulation can cascade through these networks to restore normal cell fate, and what do pioneer transcription factors play in establishing reprogrammed chromatin states?
Sure. So actually identifying the master regulators, it requires a very sophisticated network inference approaches because cancer cells, they exhibit massively rewired transcriptional circuits. So we use techniques like single cell RNA sequencing combined with computational methods like SCNIC, that is single cell regulatory network inference and clustering to map the regulatory networks in individual cancer cells. So single cell RNA sequencing has actually aided us to get a better insight into cancer cells epigenetic conditions. Also, the key insight is that not all transcription factors are equal. So the master regulators sit at a very critical network nodes where the modulation can propagate through thousands to hundreds of downstream targets. Now we have developed algorithms that integrate chromatin accessibility data with the expression profiles to identify factors that are both highly connected in network and have access to their binding sites. Thus, the pioneer transcription factors are particularly powerful to map the regulatory networks in individual cancer cells. So, single cell RNA sequencing has actually aided us to get a better insight into cancer cells epigenetic conditions. Also, the key insight is that not all transcription factors are equal. So the master regulators sit at a very critical network nodes where the modulation can propagate through thousands to hundreds of downstream targets. Now we have developed algorithms that integrate chromatin accessibility data with the expression profiles to identify factors that are both highly connected in network and have access to their binding sites. Thus, the pioneer transcription factors are particularly powerful because they can bind to closed chromatin and initiate cascades of chromatin remodeling. In our work with hepatocellular carcinoma, in the recent research work on hepatocellular carcinoma, it was identified that HNF4 alpha as a master regulator whose restoration can reprogram cancer cells back towards hepatocyte-like states, gives us a very promising insight into this concept. Also, HNF4 alpha acts as a pioneer factor opening chromatin and liver-specific regulatory elements and recruiting cofactors that establish normal hepatocyte gene expression programs. But the challenge is that pioneer factors often require specific cofactor complexes to function properly. Now, we have found that successful reprogramming usually requires modulating multiple components simultaneously. So the pioneer factor plus the pioneer factor plus its cofactors, key cofactors. Now, this is where systems level understanding becomes very crucial because we need to identify the minimal factor combinations that can flip the cellular states. It is also currently being in discovering by the researchers worldwide that the order of factor of introduction matters enormously. So sequential introduction can lead to different outcomes than the simultaneous introduction, suggesting that reprogramming follows a specific kinetic pathways that we are only beginning to understand. Yes.
So, metabolic reprogramming is intimately connected to epigenetic states through metabolites that serve as cofactors for chromatin modifying enzymes.
Mhm.
So, how do you manipulate the metabolic epigenetic axis to support cancer cell reversals, particularly regarding alpha ketoglutarate SAM and acetyl CoA availability?
Okay, so the metabolic epigenetic axis, it represents one of the most underexplored therapeutic opportunities in cancer reversal actually. The metabolites aren't just fuel; they are regulatory molecules that directly control the epigenetic enzyme activity. And this creates a very powerful intervention point because we can potentially reprogram cells by modulating their metabolic environment as well. So alpha ketoglutarate is a very crucial because it's a required it's a required cofactor for TET family of DNA demethylases and Jumonji family of histone demethylases. So cancer cells often exhibit an altered TCA cycle flux that reduces alpha ketoglutarate availability, leading to hypermethylated repressive chromatin stage. So we have developed a cell-permeable alpha-ketoglutarate analogs that can restore demethylase activity and reactivate the silenced tumor suppressors. So as adenosine methionine availability, it controls a DNA and histone methylation patterns. Interestingly, cancer cells often show paradoxical SAM metabolism. That is, high levels in some compartments, whereas deficiency in others. So, the researchers worldwide are using targeted metabolic engineering to restore balanced SAM levels, which will in return help normalize both DNA methylation and also the histone modifications. Lastly, acetyl coenzyme A is a substrate for histone acetyl transferase, and cancer cells frequently show a very disrupted acetyl coenzyme metabolism due to altered glucose and lipid metabolism. So we have found that it has been observed via the recent studies that restoring normal acetyl coenzyme A levels, particularly the nuclear acetyl coenzyme A, it can dramatically shift the chromatin accessibility pattern towards normal cell-like states. So thus, the most exciting approach is a combinatorial metabolic intervention rather than going with the one metabolite. So we simultaneously modulate multiple metabolic pathways to create metabolic environments that favor normal epigenetic states. So this isn't just providing substrates. We are essentially creating a metabolic niche that make normal cellular identity more thermodynamically favorable than the malignant states. Yeah, that's it.
So, tumor microenvironment interaction profoundly influence cellular identity, especially of that of cancer cells.
Mm-hmm.
Do you account for stromal reprogramming and immune modulation when designing reversal strategies, particularly regarding the role of mechanical forces in extracellular matrix composition?
So tumor microenvironment is absolutely critical for maintaining a malignant state, which means successful reversible strategies must address the niche and not just the cancer cell. So cancer cells and their microenvironment, they engage in a constant crosstalk that reinforces aberrant cellular identity. The mechanical forces are particularly underappreciated in this. The cancer cells, they often exist in abnormally stiff environments due to excessive collagen deposition and cross-linking. So this mechanical stress, it activates the mechanotransduction pathways, particularly YAP and Cas signaling. Now this maintains the proliferative invasive phenotypes. We are developing approach to normalize these tissue mechanics using targeted matrix metalloproteinases and cross-linking the inhibitors. So the extracellular matrix composition is equally important. Since in normal tissues we have a specific ECM signatures that provide identity cues to resident cells. Like tumor-associated fibroblasts, they produce aberrant ECM that lacks these normal cues. So we are engineering biomimetic matrices that provide mechanical and biochemical signals needed to support normal cellular identity. Also, immune modulation is very critical here because immune system serves as a quality control mechanism that normally eliminates aberrant cells. But in established tumors, the immune cells are often reprogrammed to support malignancy. So it is we are developing approaches to re-educate tumor-associated macrophages and T cells to recognize and support the reprogrammed cells rather than their malignant neighbors. Also, the most sophisticated approach is coordinated niche engineering, simultaneously reprogramming concessions and normalizing mechanical properties, restoring appropriate ECM composition, and redirecting the immune cell function. This requires delivery systems that can target multiple cell types within tumors while maintaining spatial and temporal coordination. So this could be a approach.
Okay, Dr. Pathak, thank you for your comprehensive analysis. And to the listeners, please tune in for more episodes on our podcast. Thank you.